KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
In this video, so we are discussing Apomixis and one more is Polyembryony. Important concepts. So my dear students, so in this video we are discussing Apomixis and Polyembryony conditions. Okay. Now Apomixis, first one let us start to discuss Apomixis. Apomixis is the production of seeds without fertilization. So one more time, Apomixis. So what it is? Apomixis is the production of seeds without fertilization. So means so without involvement of fertilization. Suppose if the seeds of formation taking place, so such kind of a process we can be taken as Apomixis. Now the normal process of a cell cycle involving in meiosis and fertilization is called as amphimixis. Means normal process, means by the process of like first of all meiotic division, later fertilization. So likewise simultaneously, suppose if the seeds of formation taking place means, so such kind of a process we will be taking as amphimixis. Okay, means the normal process of cell cycle involving in meiosis and fertilization is considered as amphimixis. Apomixis means once you say that is the production of seeds without fertilization. Suppose without fertilization, suppose if the seeds production, if we are observing that kind of a phenomenon, we will be taking as apomixis. Next one, amphimixis is a normal process. Means so in both reproductive systems, so first of all meiosis process takes place. Once the process of meiotic division is completed, fertilization process takes place. So that kind of entire process, another terms we will be considering as, or we will be taking as how amphimixis. Amphimixis. Next one, there are several ways. There are several ways of development. So there are several ways of development of apomictic seeds. There are different types of methods, or there are different types of ways are there. So in once you see, there are several ways of development of apomictic seeds. So there in some seeds, the diploid seed cell is formed. In some seeds, the diploid seed cell is formed without a reductional division. So means one more time you see there. So the development of apomictic seeds. For the development of apomictic seeds, there are several ways are there. Not one or two or not three or four. So many types of ways are there. So first one, once let us see. I didn't give any number, but let us discuss one by one. In some seeds, for example, the diploid seed cell is formed without a reductional division and develops into embryo without fertilization. So means here the diploid seed cell is formed. Means here the diploid cell is formed without reductional division and develops into like embryo without fertilization. Means here there is no chance to the fertilization and there is no chance to the reductional division. Next one, in some plants, new cellular cells, means new cells are there. So these new cellular cells are surrounding the embryo sac. So means when these new cellular cells are surrounding the embryo sac, automatically start dividing, protrude into the embryo sac and develops as or develops into embryos. So means here new cellular cells. So this one we see this point is there in our textbook. So means in some plants, for example, what is happening? New cellular cells or penetrating means embedded or protrude means so protrude into the embryo sac. So thereby here what are the new cellular cells are entering into the like embryos or entering into the embryo sac. So later all these protrude structures means what are the structures are entered into the embryo sac, all are converted into embryos. Next one, some apomictic plants, for example, citrus, cactus family members. Next one, Parthenium species or Parthenium members, etc. Apomixis provides a possibility to the indefinite multiplication of specially favorable biotypes without losing their productivity due to segregation. Once that's why, so this particular point we have discussed half an hour time. So in a previous classes as well, taking class, so in presence of you I have given this particular bit. So there, what is the point now we have been discussed? So means in comparison of once I have given in comparison of hybrid seeds and in comparison of apomixis seeds. So I asked to give reference to the farmers to use only apomictic seeds. Why? Because so what are the good characters are present in the apomictic seeds? All these characters will not be segregated during the process of segregation. But suppose here if you take a hybrid seeds, so even though these hybrid seeds are having good characters, while the segregation process is taking place at the time, chance is there for the segregation of good characters. So means when the character suppose segregated means in the next generation we could not see the presence of all good characters in one seed. That's why my dear students, try to remember one important point. What is that? In comparison of apomictic seeds and hybrid seeds, better to use apomictic seeds. Why? Because apomictic seeds, once you see they read this particular line, you will understand. So apomictic or apomixis provides possibility of indefinite multiplication of specially favorable biotypes without losing their productivity due to the segregation. Means here 100% no chance to the process of segregation. And so what are the good characters are there, or what are the desirable characters are there? All these are all we can multiply. How many times? Indefinite. How many times we would like to do? So that many times definitely we can multiply, and we can produce how many number of offsprings we would like to get from them. Means here we no need to bother about one particular point, that is segregation. Why? Because in apomixis there is no chance to the segregation, means there is no chance to the meiotic division. When the process of meiotic division, no chance for that particular process, so here no chance to the segregation of what are the characters are present in the parents, all these characters will not be segregated. Next one, this is somewhat information related to the apomictic seeds. Next one, polyembryony. This is important, but vast information is there. So maximum I have skipped, but remaining information I have given to you. Okay, now polyembryony. The word itself giving information. Polyembryony. Poly means many. Which one is many? Embryos. So polyembryony means here once in our textbook information point of view, I have given a definition also, that is polyembryony means occurrence of more than one embryo. So here also they have given like that only. Let us see. Is the state of occurrence, means polyembryony is a state of occurrence of more than one embryo in a seed. So means in this particular case, occurrence of more than one embryo. When means here the occurrence of more than one embryo in the seed. So means when the seed consists of more than one embryo, that particular statement we can be taken as polyembryony. So in this particular question, best examples, so where we can be taken, or what are the examples here we can be considered, means one is onion and one more is groundnut. Onion and groundnut plants, best examples. So for the polyembryonic condition. Now it was discovered by, it was means what? Polyembryony. The polyembryonic condition was discovered by Leeuwenhoek. Anton van Leeuwenhoek scientist is there now. So that particular scientist only discovered the polyembryonic condition where in citrus. This is important. So NEET examination point of view, I seen this one in some other question papers. So this particular point also you try to save in your mind. Polyembryonic condition was discovered by Leeuwenhoek in citrus species. Next one, polyembryony may be formed by cleavage of a zygote. So they are giving hints, they are going to enter into the deep information now. Polyembryony may be formed by cleavage of a zygote or the formation of embryo by the cells of the embryo sac other than the egg cell. Very clear information once you see there. The polyembryony definitely is formed. So that is maybe by chance, that is maybe by the cleavage of zygote, means after fertilization. So suppose what is a zygote is there now. Suppose if that particular zygote involves in the development of many embryos, chance is there. Or what are the cells are present in the embryo sac, means what are the cells we can see, like 2 synergids or polar nucleus and 3 antipodals, but here so chance is there from these these kind of cells except from the except then the which one, once you see, except other than the egg cell, means here this particular egg cell is not involved, some other cases only. Okay, means once you see this statement one more time, polyembryony may be formed by the cleavage of a zygote or the formation of embryo by the cells of the embryo sac other than the egg cell, means here egg cell is not involved in a formation of a polyembryonic condition. Then usually what we can be considered, sir, means here answer is there, usually synergids. Usually synergids are definitely involved in the development of embryos or polyembryonic condition, or by the development of more than one embryo sac within the same ovule. Or suppose like so many number of embryo sacs are there, means again what are the cells are present in the so many number of embryo sac, there again synergids, antipodals, next one remaining cells except egg cell, all cells will be involved in involved in the development of polyembryonic condition in one seed. Why? Because the same ovule, means after fertilization this ovule converted into seed. That's why I have used the term as a seed. Means, so here one point, polyembryonic condition, we can see the development from the zygote, or what are the cells present in the embryo sac, other than the egg cell, definitely all cells will be involved in the development of embryo, or polyembryonic condition. So generally what we are studying means our subject point of view, till now our scientists observed this one. So mostly synergid cells are involving in development of polyembryonic condition. So that is a main important point in this class we are discussing. Next one, polyembryony is a practically important. Once you see there again, they have given this point, polyembryonic condition is a practically important because genetically uniform parental type seedling are obtained from the nuclear embryos. One more time, polyembryonic condition is a practically important because why it is practically important? Genetically uniform parental type seedlings are obtained from the nuclear embryos. So here when genetically uniforming parents we will be taking in seeds or seedlings. So already these seedlings are having, so what are the parents characters are there? All these characters will be present in the offspring. So thereby definitely in polyembryonic conditions also, so what are the parental characters are there? All these parental characters only are transporting to the next embryos. So definitely these embryos or these seeds definitely involves in the propagation or involves in the which one increasing of its life cycle. Okay, so likewise so polyembryonic condition we can take. Next one, apomixis condition also we can discuss. Next one here, what is importance or what is information? Next one, most important theory cause. Most important theory cause polyembryony. So most important theory cause polyembryony. So most important theory of cause of polyembryony is neurohormone. Okay, neurohormone. So one more time you see there, most important theory of cause of polyembryony is neurohormone theory. Amma, 1 minute, amma. Neurohormone theory. That neurohormone theory was given by Haberland. Amma, one more time. Most important theory of cause of polyembryony. So means likewise, what are the causes of biodiversity? Likewise, we see here even. So what is the important or main cause of polyembryony? So that one theory is there for explanation of that one. That is neurohormone theory. That is neurohormone theory given by Haberland. Is important point. So Haberland has given neurohormone theory. So for the which one? Polyembryony condition. Next one. Once you see there are different types of like a polyembryonies are there. So there first one is cleavage. Once let me give you the side headings. Later let us discuss one by one. Cleavage polyembryony, simple polyembryony, mixed polyembryony, and adventive polyembryony. One more time. Cleavage polyembryony, simple polyembryony, mixed polyembryony, and adventive polyembryony. First of all, side headings try to by heart try. So means when you will be having all side headings in your mind, so thereby information easy the varam. Okay, ma? So suppose if you don't know side headings itself, then where you will say this particular information under which one? So that's why first of all headings alone panikonga, apro information konchu try save panikonga. Right. Now you see there. Cleavage of polyembryony. The word itself giving cleavage means what? Breaking only no. So that breaking is there or not, now let us see. So due to cleavage of a zygote is there, ma. So due to cleavage of a zygote or proembryo. So zygote first formed product, or after somewhat development, when zygote is going to be developed, proembryo develops. So that particular part only cleavage. So into how 2 or more embryos and split part developed means here what are the cleavage particles are there, no? Emma. So if zygote is there, adi segment size. Cleavage means that is only break. So all breaked parts, split parts. Emma. So LMA split parts develops into embryo. Point you understood here, zygote or proembryo. So split, what are the particles or what are the like splitted particle parts are there, no? All parts are developed into embryos. So thereby we can observe the presence of more number of embryos. So next one, this type of polyembryony is common in gymnosperms. Means here zygote splitting or zygote splitting or proembryo splitting. Likewise, such kind of development generally we will observe in gymnosperms than the angiosperms. So means in comparison of gymnosperms and angiosperms, where we can observe. Though where we can observe this kind of phenomenon means we can observe very more in gymnosperms than the angiosperms. Which one? Cleavage type. Next one more you see there. So erythronium. So here examples of like angiosperms I have given: erythronium americanum and nymphia and crotalaria. Erythronium, nymphia, and one more is crotalaria. So one more time, erythronium, nymphia and crotalaria members, if you see there, no, this kind of a cleavage polyembryonic condition we can observe. So these members are belongs to the angiosperms. Showing cleavage polyembryonic. Now let me move forward to the second one, that is a simple polyembryony. The word itself giving, so there is no any difficult, simple. Due to the presence of more than one embryo, due to the presence of more than one embryo sac. Okay, so when more number of embryo sacs are there, no, definitely we can find out presence of more number of embryos. means egg cells. So definitely from that egg cells to again definitely we can see the development of more number of embryos. So thereby when more number of embryo sacs are there, now due to the presence of more number of embryo sacs, we can see the presence of more number of oospores or egg cells. So we can see this kind of simple embryony in brassica members. Next one, mixed polyembryony. Which one? Mixed polyembryony point of view. More than, இப்போ mixed means எல்லாமே male female எல்லாமே வரும் இப்போ பாருங்கம்மா. So more than one pollen tube entering once you see there mixed polyembryony condition point of view. இப்போ when more than one pollen tube entering into the ovule or entering into an ovule and fertilizing with synergids and antipodal cells. Once you see more. So means here there are different types of pollen tubes are coming. So which are moving towards the ovule, and it is not fusing with the egg cell, fusing with synergids as well as antipodals. So thereby, so definitely, so we can see the development of a polyembryony condition, mixed one. Okay, next one, adventive embryony. Adventive embryony, diploid nucellus. இது கொஞ்சம் இம்பார்ட்டன்ட் அம்மா. ஃபர்ஸ்ட் ஒன்னும் லாஸ்ட் ஒன்னே கொஞ்சம் இம்பார்ட்டன்ட், மிடில் ரெண்டுமே ஈஸி தான். Now diploid nucellus or integument cells from embryos means here once you see there diploid. So what are the diploid nucellus or the diploid embryo sacs are there? No, all these are all definitely involves in the development of embryos. Means here these are all. So means here the nucellus cells or integument cells form embryos. So means here what is the main important point we have to be discussed means, so here in adventive polyembryony diploid nucellus or integuments, இப்போ diploid nucellus or integuments form embryos. Means so suppose if embryos are formed from the integuments or diploid nucellus, so such particular polyembryonic condition we will be taking as adventive polyembryony type. So this kind of a phenomenon, so where and where we are observing means we will be observing in a citrus, Opuntia, mango. So there are different types of examples over there. So in all these examples, so we will be discussing means citrus is there, Opuntia is there, Mangifera indica, mango also is there. Thereby here in adventive embryony nucellus involves in the development of embryo as well as integuments also are there. Next one, if up to here up to Mangifera indica, mango are the different types of polyembryony comes. Next one is separate information. Now if extra embryos, one more time, once you see, if extra embryos develop from same embryo sac, இப்போ extra embryos means actually suppose from the embryo sac one embryo chance is there, but suppose if extra embryo comes, suppose if extra embryo come from the same embryo sac means that kind of that one we will be taking as true polyembryony. True polyembryony means when we can be taken, suppose from one as embryo sac, suppose if we are observing more than one embryo formation, so that kind of embryo formation we will be considering as true polyembryony. Next one more time you see, if embryos are developed elsewhere, suppose ஏதோ ஒரு particular part involves in the development of embryos means, so that kind of a phenomenon we will be taking as a false polyembryony. Point to understood. So means here other than the embryo sac, either one particular part means elsewhere. So either one particular part definitely involves in the development of embryos means, so that kind of polyembryonic condition development we will be taking as false polyembryonic condition. So this kind of false polyembryonic condition, so we will be observing. So once you see that Balanophora, Rhizophora, Balanophora, likewise parasitic plants we see now, so that is now in a Balanophora an extra embryo development from the endosperm. Once you see, so here embryo development is there, Embryo development is not from the any part of the embryo sac, it is developing from the endosperm. Next one, let us discuss advantages of having of seed in angiosperms. Means seeds structures mostly we will be observing, we will be noticing in angiosperms only, no? That's why once let us discuss what are the advantages are there with the seeds. Once let us discuss, there are 4 points are there, let us discuss and let us leave this. Now once you see, seeds can remain dormant for many years and germinate on in turn of favorable conditions. Means here, so seeds can sleep means by reducing its metabolic activities, so they will be reducing the metabolic activities and they can go with an inactive metabolic state is known as dormancy. We know very well, we have learned in the second chapter. So means dormancy is nothing but when the particular plants or the particular plant parts reduce the metabolic activities and go with a state of inactive state or the rest of inactive. So that particular phenomenon or term we will be taking as dormancy, that is also for many years. Once we have seen now. So Phoenix dactylifera 2 years of dormancy we seen, and Leucas and Leonotis we have seen 10,000 years of dormancy. Try to remember. Okay. Next one, and all these are all having capability and to form or to germinate in favorable conditions. Later, reproductive process is not dependent on water. So here one more important point, this particular reproductive process, what are the processes and everything are there, no? All these kind of processes here not depending on this water. Why? Because angiosperms well developed one only, no? So reproduction process and everything not depending on the water. Means up to pteridophytes, definitely plants depending on the reproductive depending on the water for the process of reproduction. Next one, seeds have sufficient food resource to initiate embryo development. If seeds definitely seeds are there, means definitely we can find out presence of endosperm. No, presence of endosperm if we are observing means definitely which one will be there? Nutrition will be there, or the availability of food materials are there. So when seeds are full and fully filled with food materials, no, definitely those food materials helps for the embryo development. Next one, hard seed coat protects the seed from the environmental extremes or environment extremes. Means here because of the hard seed coat, means when particular seed coat is active or like a very hard, no, so due to that one it can it can overcome the unfavorable conditions. Unfavorable conditions, and it can form or means it can germinate in favorable conditions. Okay. Next one, the seeds have a better adaptive strategies for dispersal to the new habitats. Means if you see these particular seeds, no, so some kind of seeds are definitely having better adaptive strategies. Definitely they will be having good strategies. So because of the having of different types of mechanisms, so definitely they can dispersal or they can get a dispersal from the one habitat to new habitats. So likewise, so author has given a different types of uses of seeds. So I will not take more than this one. So but in this video, so we have discussed regarding apomixis, polyembryony and polyembryony types, cleavage, simple, mixed and adventive, and later we have discussed the information of advantages of seed. So revise it once. Maximum information you can save in your mind. Suppose if you are feeling burden, no, so don't read like this one, cleavage examples, and from where polyembryony formation, and likewise and simple from where, and mixed from where embryo formation, and in adventive from where embryo formation. Likewise, just in 1 or 2 sentences you try to learn the information. So when we will start to prepare the Neet examination, now at the time we will start this entire information by one more time. So take care and be safe at home.